This is part 2 of the chapter 9, section A, PowerPoint slides. So we left off, we were discussing skeletal muscle fibers, and we left off with sarcomere, which is an individual contracting unit of muscle. One sarcomere, as you can see here, extends from one Z disc or Z line to another Z disc or Z line. The I band spans the distance that's taken up only by actin, which is the blue filaments. So I bands are half belonging to one sarcomere and half of the I band belongs to another sarcomere. The sarcomere actually starts in the middle between one I band and travels the distance across a single A band to the middle of the next I band. So that's a sarcomere. From Z disc to Z disc, the I band is the region that is light on the striations on your muscle are formed because the I band is light, because it's only composed of the thin filaments or actin. And the A band is dark because it is composed of myosin, which overlaps actin, but myosin are the thick filaments, which are red. They're shown as red in this illustration. The H zone is the region between actin, so that means it only contains myosin. And then there's an M line, which is in the middle of the H zone. Okay, so myofilaments are an arrangement of actin and myosin within a sarcomere. The actin myofilaments are called thin filaments. The actin and the myosin together are going to produce your muscle contractions. So the myosin filaments are the thick filaments. And you can see here they have kept, in the drawing, they have kept the actin or thin filaments blue. And they have kept the myosin or thick filaments red. These are your myosin filaments. And so you can just see all the parts of the sarcomere blown up from Z disc to Z disc. You can see the I band, which is formed only of actin, the H zone, which is the area that only contains myosin. The M line is the middle of the H zone, and then the A band is the dark colored striation in the muscle fiber because it contains myosin, which is a thick filament. And then we see another filament called titin. Titin are these little corkscrew yellow filaments, and what they do, titin connects the actin to the Z disc, connects the actin filament, or the myosin filament to the myosin and the actin filament to the Z disc. Okay, so it helps connect between sarcomeres, basically. Okay. The myosin and the actin are going to form cross bridges. They're going to connect with each other during a muscle contraction. There are two proteins that you do need to know about when we study actin or the thin filaments. Actin is made up of G-actin, which is globular, and F-actin, which is like a rope, and two F-actin strands twist together and form a twisted rope to form the thin filament. So it's F-actin that forms the thin filament, and G-actin is a subunit that's going to contain an attachment point for myosin. Now, two other proteins are regulatory proteins, and they are tropomyosin and troponin. So this is showing you the composition of the myosin filaments. They're going to contain two binding sites, a binding site for ATP and a binding site for actin, which are both going to be involved in the muscle contraction. The actin is going to contain the F-actin, which is the rope-like strands of actin that are twisted together to form that thin filament. The tropomyosin is wrapped in such a way that it covers over the myosin binding sites on the actin. So each actin has a globular G-actin subunit that has a myosin binding site on it, and the tropomyosin typically covers those binding sites up, and the troponin holds the tropomyosin in place. All right. Then we have titin, which holds the thick filaments in place. We have dystrophin, which links the thin filaments to the sarcolemma, which of course is the plasma membrane of the muscle fiber. And we have other proteins like nebulin, myomycin, C proteins that bind filaments or sarcomeres together and maintain the alignment of the sarcomere. Dystrophin is involved in, dystrophin, remember, links actin filaments to the sarcolemma. It's going to create stability between the thin filaments and the muscle fiber itself. In Duchenne muscular dystrophy, DMD, it's the most common form of muscular dystrophy. It is a sex-linked recessive disease, so it's almost exclusively found in males. And what happens is that the gene for producing the protein dystrophin is defective. And so the protein that links thin filaments to the sarcolemma, helps stabilize the sarcolemma, is defective. So the sarcolemma tears easily, which allows excess calcium to enter the muscle fiber, and it will damage the muscle fiber. And that can lead to a drop in muscle mass over time, which is going to lead to falls, and it's going to lead to falling down and just lack of coordination. It does start in the extremities, but it does move to it's a progressive disease, so it does move to the chest muscles and cardiac muscle, which of course is going to just continue to progress. But with good care, supportive care, these patients can live into their 30s and beyond. And I know several personally who within the community college system who have lived well beyond their 30s and are still going, very strong. So definitely with good health care, a person with Duchenne muscular dystrophy can have a good life and a long life. The sarcoplasmic reticulum is the endoplasmic reticulum. It's the smooth ER of the muscle fiber. It functions in regulating calcium levels, and it also stores calcium. And we're talking about calcium ion here, which has a +2 charge. The calcium ion is going to be really important in the muscle contraction. It's going to play a role in the muscle contraction. T-tubules are formed from the sarcolemma. So basically the sarcolemma sort of protrudes deep into the interior of the cell, and it allows nerve transmission to reach into deep into each muscle fiber. A triad is composed of the terminal cistern of one sarcomere, the T-tubule in the middle, so that means the terminal end of one sarcomere, the T-tubule in the middle, and then the terminal end of a neighboring sarcomere. So triad because it's two terminal cisternae and one T-tubule. The T-tubule allows voltage sensors to carry electrical current deep into the muscle fiber, which is going to trigger a muscle contraction. And then the sarcoplasmic reticulum proteins are going to control the opening of calcium channels. And so when the electrical signal travels down the T-tubule deep into the muscle fiber, it's going to cause the release of calcium, which, like I say, will play a role in muscle contraction. The blue structures here are your sarcoplasmic reticulum. And then you can see a triad, which is composed of a T-tubule. This is a T-tubule here, which is just the sarcolemma. It's gray like the sarcolemma. It's just the sarcolemma, which has formed, it's folded and formed a tube way down into the muscle fiber. So there's a T-tubule and then the terminal cisternae of the sarcoplasmic reticulum that neighbor each other, that are side by side. So this is one terminal cisternae and this is another terminal cisternae. The terminal cisternae comes together here meets at the T-tubule, and that gives you a triad. Contraction is defined as the activation of cross bridges to generate force. So we're going to learn what these cross bridges are. We know they're formed by actin and myosin. And contraction is going to result in shortening of the muscle. We use the sliding filament model of contraction, which states that during contraction, thin filaments slide past thick filaments, causing actin and myosin to overlap even more than they already do. None of the filaments change length. They just overlap more. They come, basically the actin filaments come closer to each other. The sliding filament model of contraction, again, causes shortening of the muscle fiber. It causes the Z disc to be pulled toward the M line, the I bands to shorten, the Z disc to come closer together. The H zone will disappear. That's the area where it's just myosin. So what will happen is the actin will slide over the myosin, and the two actin filaments that were separated from each other will now almost touch, or they will touch. And the A bands will move closer to each other as well. So this is fully relaxed, and you can see there's an H zone in the middle of the A band. There's an area where actin overlaps with myosin, like right in here, but it doesn't overlap completely with the myosin. Then a contracted sarcomere brings the two actin filaments together. There is no more H zone, and the two actin filaments totally overlap with the myosin filaments. So it's really just the actin and the Z lines that are moving in a muscle contraction. The myosin stays still. The decision to move, and we're talking, of course, about skeletal muscle here. The decision to move is activated by the brain, and then the signal is transmitted down the spinal cord to motor neurons, which then activate muscle fibers. So neurons in muscle cells are excitable cells. They're capable of action potentials or electrical impulses. And the action potential crosses from neuron to muscle cell through release of the neurotransmitter acetylcholine, or ACH. And this is going to kind of lead into our nerve chapter as well. So there will be a little bit on neurons and neurotransmitters, because you can't really have a muscle contraction without a nerve impulse. So we have to learn both. Ion channels that are located within the plasma membrane of your neurons and your muscles play roles in changing membrane potentials. Now, membrane potentials are basically voltages, and a voltage can have an overall negative or an overall positive charge. But At a resting state, the voltage across the plasma membrane is negative.